right. 8.1, identifying DNA as genetic material. So Mendel in the 1800s knew that there was something that was passed on to kids, that parents passed on to kids, that gave them traits. But he didn't know what that material was. So he wasn't sure. He knew it was something that was physical, but he didn't know what that physical thing was. And they didn't know for about 100 years after Mendel's experiments. But in the early 1900s, 1920s, up through the 1950s, they did a lot of experiments to try to determine what this thing was. In the 1950s, they finally identified it as DNA. So DNA was identified as genetic material through different experiments. And this proved that this was the substance that was passed on from parents to kids, which gave the kids the traits that parents had given to them. And then that picture shows you a mouse. Did we talk about this before? About why the mouse is glowing? Yeah, they did this with cats too. Did I tell you about the cats that they've done with, they made glow-in-the-dark cats? Same thing with the mouse. Remember how they did that? How do they make cats glow in the dark? That's right, jellyfish DNA. Yeah, good. Because jellyfish glow different colors. And so that's what they did. They took the gene from a jellyfish, which made it glow in the dark, and they transplanted it into a cat. And that's the amazing. Same with this mouse right here. They did the same exact thing. They took a gene from the jellyfish, put it in the mouse, and now they had the gene that can make the glow in the dark. Okay, so early 1900s, they knew there was a substance that was passed on, but they weren't sure what that substance was. They narrowed it down to two choices, though. Is either DNA or protein. You don't need to write down these, there's too small for you to see anyway, but these boxes, don't worry about those boxes right there. I just want you to get the main idea on here. All you need to really do, you can draw it if you want to, but you don't need to. The main thing I want to show from this picture is that they thought it was protein and not DNA, because protein could make a lot of different shapes. There was 4 basic shapes that protein could make, and there are a lot of possibilities from those 4 shapes. DNA, though, only just has this double helix. So DNA to them was just too simple. They thought that, DNA only had a few different chemicals in it. It was very repetitive. It repeated itself over and over again. Well, protein had 20 different chemicals compared to 4 for DNA. So because there is 20 different chemicals that makes up protein, and you had a lot of different shapes that you could make with protein, they assumed protein must be the genetic material that's passed on. But they weren't for sure just yet. But most scientists at the time thought it was protein. But DNA was still a possibility. And so they were trying to figure out which one it was. So the main thing to say from here is that, this heading right here, they weren't sure. Was it DNA or protein that makes up the genes? There's a lot of debate about this. And they did experiments to show which one it was. We'll go through the experiments today. So Griffith experimented with bacteria that causes pneumonia. And he was able to narrow it down from his experiments on whether it was protein or DNA. So it was in 1928 when he did these experiments with bacteria. So he looked at 2 different kinds of bacteria. You have smooth bacteria on this side and rough on this side. So rough bacteria is called rough because the colonies it makes are little dots and looks kind of rough. Smooth bacteria makes bigger colonies. Yes.
I did a science experiment in 5th grade, and we were using petri dishes like that. And I had a lot like the isolate is like fungi. Is that also bacteria, or is that?
Some of it could be fungus. So there is a lot of fungus in the air too. What we call mold is usually fungus, but there also is bacteria in that as well. So it is a combination, I'm sure, of both bacteria and fungus. Bacteria that looks like this, fungus looks more fuzzy generally. So we looked at two kinds of bacteria, the S form, and it was called S because S is smooth. And so the S form is this side right here. That was deadly. And then the R form, called R for rough, on this side, that was not deadly. So he would inject these bacteria types into the mice. And he put the living smooth bacteria into a mouse, it died. He put the living rough bacteria into a mouse, it survived. But then he took the bacteria, the smooth bacteria, and he put it about 140 degrees Fahrenheit. And that was enough to kill the bacteria. And he knew that that's what he was doing, was killing the bacteria. And then he put it into a mouse. And the mouse survived. And so he confirmed he had killed the bacteria. With dead bacteria, the mouse was just fine. But what happened when he put dead smooth bacteria in with living rough bacteria? The mouse died. And that really shocked him. Because what was the living bacteria? It was the R, which should not have killed the mouse. The living R bacteria, was not deadly. And the smooth bacteria was dead. So he didn't understand, how could this happen? How could you have mixed the dead bacteria that was deadly with the live bacteria, which is not deadly? How would you get a dead mouse? Well, he said there must have been some chemical that the dead bacteria transferred to the living bacteria and converted them or transformed them into harmful bacteria. So he called this a transforming material. There must be some kind of chemical that he called a transforming material, which passed from the dead S bacteria to the living R bacteria and transformed them into S bacteria, which made them deadly. And this is why bacteria is so hard to treat, and why we get so many different kinds of bacterial infections, like staph infection is very hard to treat. Antibiotics are constantly having to be, we need new and new antibiotics because the ones we have over time just wear out and don't work very well. Because bacteria are constantly mutating. Oh, sure. And they're constantly mutating and adding genes to other bacteria. So what happened here? Well, the dead S bacteria transferred their genes to the living bacteria. And the living bacteria got those genes that made them deadly. And that's why the mouse died. And at the time, he didn't know if that was DNA or protein, but he knew there was some kind of chemical that had been passed over. And it's really interesting. We don't know how this happens, but the living bacteria can somehow sense which other bacteria need a gene. And they can somehow sense which ones don't have a gene that helps them. And they'll make a tube and transfer the gene through the tube to the bacteria who need the gene. We don't know how they can do that. They don't have brains, they don't have eyes, but somehow they can sense which ones need a certain gene and they pass it to them through a tube.
So was it the dead thing that did that?
Yes, and so the red bacteria that was living, which is not harmful, just picked up these genes. It basically tore apart the bacteria and pulled out the genes it needed. Did you have a question?
Oh, I was just gonna ask.
Yeah, so the red bacteria in this case was able to, basically, bacteria eat bacteria. Bacteria will pull apart bacteria. And so the red bacteria just pulled apart the blue bacteria, but was able to recognize a gene that they needed, and it pulled the gene in to their body. And that gene became part of their nucleus, which now they became like the blue bacteria, which was deadly. So Griffith found living bacteria that was S in the sample. So the R bacteria had converted into S. They had become S protein. So by taking the genes from the dead S protein, those R bacteria had become S protein. They had been transformed from R into S. So Griffith called the material that had changed R into S the transforming principle. And again, nowadays we know it's DNA, but at the time he didn't know what this was. Did everybody have this one down? Now, he was a little bit fortunate in his experiment because DNA denatures or breaks apart 190 degrees Fahrenheit. And so he was able to have a temperature, 140 degrees, which killed the bacteria, but he didn't get above 140 to 190, which would have killed not killed DNA, but would have denatured it to the point where it would be usable. So if he had put it in an oven at 200 degrees Fahrenheit, he would have killed or torn apart the DNA, and he would not have discovered this. So he was fortunate that he only used a temperature that was enough to kill the bacteria, but not enough to destroy the DNA. Another scientist a little bit later on, Avery, he isolated and purified the transforming principle. So Frederick Griffith, he said that there is a transforming principle, but he was not able to isolate it and figure out what it was. But other scientists at the same time were able to take out that chemical and figure out what this was. So Oswald Avery, he was able to isolate and purify the transforming principle, which the R bacteria was used to then transform into S. And he took that transforming principle and he gave it to R bacteria to confirm that that is what really did change R bacteria into S. And he showed that that is true. This chemical he was able to isolate did transform those R bacteria into S. So now that he can confirm that he had the right chemical, he now needs to know what the chemical was. And so he did 3 tests to figure out what this chemical was. So the first test he did was qualitative. Remember we talked about qualitative data and quantitative data? Qualitative data tells you, quality without a number. And so it can tell you if something is present, but not how much. And so this test just tells you if there was DNA present or not. So qualitative would just say you do or you don't have DNA. It won't tell you how much DNA is present. It just told them that there was DNA present. So that was the first test that helped to confirm that this was DNA. But you need to do more tests to know for sure what he was dealing with. So then he did some chemical tests, which you can see in the chart. That chart, don't worry about writing down all those things. You don't need to do that, but just note that that's what he saw in his experiment. He was able to look at the chemicals that were in there, and he found nitrogen and phosphorus. And what he found is that it was the same ratio of nitrogen and phosphorus that you see in DNA. And so this was very good evidence that he was dealing with DNA because the chemicals that made it up were the exact same as what you find in DNA. So these are his samples. These are the transforming principles that he isolated, and he compared it to the known sample for known values for DNA. And you can see the numbers are very, very close to what he should have had for DNA. Did you have a question, Lucy? Okay. And then you can see the ratio was also very close to what you had for DNA. The last thing he did was an enzyme test. And he put in these enzymes that destroyed DNA into the transforming principle. And if he did that, then it didn't work anymore. There were also enzymes that you could use that would destroy protein. And he put those in too. And the principle still worked on the mice. It was only when he used the enzyme destroying, the DNA destroying enzymes that he saw it did not work. And so these three tests combined gave very good evidence that it was DNA that was being transferred to the bacteria that made them change. A few other people came along after, about 20, 30 years later, 1952. Hershey and Chase. They confirmed that DNA is genetic material. So there, all these experiments before Hershey and Chase had pretty good evidence, but they still couldn't say 100% for sure that this was DNA that's being transferred. But Hershey and Chase were the ones who actually could confirm it. So Alfred Hershey and Martha Chase studied viruses that infect bacteria. Bacteriophages are viruses that kill bacteria. And that's what we see on here. Actually, don't worry about that one. So basically, this is a cell surface right here. So you can see that compared to the bacteria, they're very, very small. So these are the bacteria back here. You can see kind of look like a hot dog. They're just cylinder shaped. And compared to the bacteria, they're very, very small.
Do we need to know about the page for?
Oh, yeah, these don't, no. Mm.
We need to stop.
Just this right here. Oh, sorry. You know what? I'll just go and delete this. It'll be easier because I don't know. I didn't mean to have these on here. So I can just put down the thing you need to know. Just that. That's all you need to know. Does everybody have that down? Oh, okay. So bacteriophages are basically, that means bacteria eater. These viruses kill bacteria. So what a virus is the protein capsule, which is what you're seeing right here. So the protein is what makes up all of this, makes up the legs on this virus. It makes up this capsule. The DNA is held in this place right here. And then when it is on the bacteria surface, you would see a needle and that would go in, these needles right here that go into the bacteria. And so it actually opens up the cell membrane at that point. And at that point, it just injects the DNA in. So it's kind of like a shot. I mean, it's very similar to that, where you have a needle and it injects viruses' DNA into the bacteria. And at that point, it kills the bacteria. Yes.
How does it move around those stuff to move forward?
Oh, that's a good question. They can move a little bit. I mean, they're kind of like a machine. So they do have some parts, like the needles that can move. So this part can move up and down. Like it can do that because it has to touch this part to the cell surface in order to put in the membrane. All right, guys, lectures, in class is how you learn. And so I don't want to see you having religion books out. I don't want to see you working on homework, guys. This is time for you to learn biology. You have plenty of time in the school day. You have study halls. There's plenty of time for you to do homework elsewhere. But this is time for biology class, guys, okay? So this right here can move up and down. And this can move in and pierce this area like a needle. But they don't swim. They don't really walk. So most of the time you get viruses by touching a surface. That's why they say, wash your hands a lot. So viruses are mostly just staying in place. Like if somebody sneezes, a virus can go out of your nose onto like the tabletop, but the virus is not going to crawl to you. You have to touch the tabletop and then touch your nose to get it into your body. So they're pretty limited in mobility. They're usually picked up by cells and that's how they're spread. But when they are on the cell, they can then bounce down and pierce the cell with the needles. That's about all they can do, really. They're limited movement. So they're a little bit like a machine, but they can't do a whole lot.
So if we touch it, you have to put it to your face.
You have to have an opening, so you have different openings, like your ear, your eyes, your nose, your mouth. There has to be some opening for them to come in because a virus on your skin can't get into your body. Your skin is waterproof. It protects your body very well. So you need to have, now if you had a cut, that's different. So if you had an open cut, you can go in that way. But besides that, you have such an open area for you to get the virus into your body. Ears are a really common way to get the virus. People don't really think about ears being an opening, but they are. Eyes also are an opening we don't really think about. And of course, mouth and nose.
So it can't get in like your pores and stuff.
Yeah, I mean, your pores are just so small, and below the pores, it is closed off. So it's very, very difficult for bacteria and virus to get into your body through your skin. It's unless you have an open cut, they really can't get in that way.
Can COVID get through your ears?
Can what get into yours?
COVID.
COVID, yeah, it can. Yeah. So like your earbuds would be a way to transfer COVID. Or if you touch, a table and then touch your ear, that could be a way to transfer COVID too. So we only, or your cell phone, if you were to put your phone up to your ear. We don't really think of it that way, but you can definitely transfer it. You can get sick very easily by having contact with your ear. So what they did to determine, because viruses were a really good way to determine whether it was DNA or protein being transferred, because that's what a virus really is. A virus is really just protein and DNA. So they can isolate the protein versus the DNA in a virus and see which one is being transferred. So bacteria has a lot of sulfur in it, but there's no sulfur in DNA. So they had the bacteria infected with a virus that had sulfur that was radioactive. That sulfur is only found in protein. So the protein on the virus had sulfur, but no sulfur in the DNA. And they had the virus then infect bacteria. Well, if the protein was being transferred from virus to bacteria, they should find that sulfur in the bacteria, and they didn't. So that confirmed that no protein was being transferred, that the bacteria were not picking up the protein from the virus. Then what they did is they used radioactive phosphorus. Phosphorus is only found in DNA, and they made that radioactive. And now they looked for radioactive phosphorus in the bacteria, and they found it. And so this confirmed once and for all that DNA is what's being transferred to the bacteria and not protein. So they finally knew for certain that DNA is genetic material that's being transferred on, whether you're talking about a virus or parents to their kids. We now know for sure that DNA is what's being passed on and gives you traits. And so Hershey's and Chase's experiment in 1952 was able to finally show that protein is not being transferred on. DNA is what's being transferred. All right, so Monday we'll talk more about other experiments they did to further see what's made up in the DNA and how it looks. This is the end of 8.1. Homework today on page 214, do 1 through 6. So for those at home on Zoom, I'll just go ahead and end Zoom today for you all. And then, do you have any questions before I leave Zoom, guys?
No, thank you.
All right,
No,
have a good weekend.
thank you.